What do humans, flies, and worms have in common? More than you might think. In fact, even scientists have been amazed to find that the genes that control early development in fruit flies and humans are essentially identical. And genes associated with heart disease in humans have the same metabolic functions as related genes in yeast. As a result, model organisms like mice have become even more important and relevant to health research.
One of the technologies that allow us to insert a mutation similar to what is found in patients into a different organism, in this case a mouse, and see whether that same change, that same genetic change, causes a phenotype, a behavior or a pathology in the mouse that is similar to what is seen in patients. And after doing that, seeing whether there are similar manifestations in a different organism, try to dissect what goes wrong inside the cells.
A mouse in which a foreign DNA, also known as a transgene, has been incorporated into its genome is called a transgenic mouse. There are two main ways to create a transgenic mouse. First, a piece of DNA can be injected directly into a fertilized mouse egg using microinjection equipment like this. The injected DNA can become incorporated into a random location in the genome. In this method, fertilized eggs are collected before the paternal and maternal nuclei have fused. At this stage in development, each nucleus is called a pronucleus. Under the microscope, a sharp micropipette is guided to penetrate the male pronucleus, and the transgene DNA inside the micropipette is injected. These eggs are then implanted into a foster female mouse, and the pups are born 3 weeks later. Not all pups may have successfully incorporated the transgene. To test which pups have the transgene, a small sample is taken from the tail and analyzed by PCR and gel electrophoresis. Mice that have the transgene are bred with wild-type mice. The offspring are again tested for the presence of the transgene. It is important to check that the transgene can be stably transmitted to the next generation in order to establish a line of mice that carry the new gene. The direct injection technique is limited because the transgene inserts in a random location, often with multiple copies, producing results that can be complicated to interpret. To create mice with specific genotypes, a method was developed that replaces a normal gene with a mutant transgene. This method exploits a phenomenon called homologous recombination, where two homologous sequences of DNA can recombine, much like in a meiotic crossing over. A mutant transgene is created with flanking sequences that are identical to the DNA surrounding the normal target gene, and the transgene replaces the normal gene.